Assalamualaikum warahmatullahi wabarakatuh. So today we would like to present about the technology in biology, chemistry and physics of dairy products, which is milk. So before that, we would like to introduce ourselves. My name is Muhammad Afidul Usman.
My name is Muhammad Saiful bin Amin.
My name is Fatin Raima binti Abdul Rahman.
And my name is Muhammad Ilham Ali bin Halim.
Now I would like to introduce about milk. Milk is a dairy milk that is extracted from farm animals during or soon after pregnancy. Milk is a very valuable food that require handling.
Okay, now we proceed to biological technology in production of milk. So before we transfer the milk into the process, we need to check the quality in the farm level. Okay, firstly, milk is sterile and colonized by bacteria before it leaves the udder. The temperature must be 35 degrees C to prevent microbial growth and rapid cooling. The test that we can use is somatic cell count test, or SCC. It is to detect the mastitis infection. The SCC level must be below 200,000 ml for good extraction of protein from milk. The mastitis caused by microorganism Staphylococcus aureus. After the quality test at the farm level, we do quality testing at milk intake. The milk will be tested before acceptance into processing train. The test that we use is Resazurin test. It is to determine the microbiological quality of milk. In this test, we use blue dye that will turn to purple, to pink and lastly to colorless due to oxidation reduction reaction of the bacteria in the milk. In biological technology also, we have separation and clarification process. Firstly is removal of bacteria, bacteriophage. It is capable to remove 99.9% of spore forming by bacteriophagy and microfiltration. Next is removal of somatic cell. Somatic cell is known also as leukocyte. This will reduce the presence of listeria that trap inside the leukocyte. It will decrease the burden of biofilms and lead to more efficient work of HPE. And lastly is milk bacteria clarification. This will avoid problem during cheese aging. It will improve shelf life and organoleptic properties of dairy products. The clarifier type is centrifugal separator.
Next is technology in biology of milk production. First is pasteurization. It uses gene modification, molecular marker technology and genomics as research tool in the development of improved pasture for more efficient production, better quality and cleaner environment. Scientists have identified ryegrass gene encoding the key enzyme involved in the synthesis of lignin, a complex organic polymer that makes plant cell wall woody. Developing genetically modified grasses with improved herbage quality by altering the lignification of cell wall. It is also using a state-of-the-art genomic technologies to identify key agronomy genes in native and exotic grasses and legumes. This program aims to improve pasture plant characteristics for the benefit of grazing industry. Next is cattle. Technology nowadays developing genomics technologies for increased genetic gain and improved milk production in dairy cattle. It's identifying specific genes and genetic markers to either modify or select for traits associated with milk yield and composition, lactation, persistency and sensitivity to nutritional status. These genetic markers identifying that code for increased production are being used by dairy producer to breed better performing cows. And at some future time, research may lead to genetically modified dairy cattle that produce milk with increased amount of casein protein, thereby increasing the value of their milk.
Moving on to the technology in chemistry for milk production. First, the raw milk need to undergo detailed analysis. Before going through the whole process, the quality test was tested on the acidity, freezing point, fats and proteins. The first one is acidity. There are two types of acidity in milk, which the first one is natural acidity due to citrates and phosphates present in the milk and dissolves carbon dioxide during the processing of milking. The second one is developed acidity. This is due to the lactic acid produced by bacteria. Because when the bacteria use the lactose in the milk as a nutrient, it will convert the lactose to lactic acid. Generally, the acidity of milk means the total acidity, which is natural plus developed, or titrable acidity. This acidity is determined by titrating a known volume of milk with standard alkali to the point of an indicator like phenolphthalein. Titrable acidity is a measure of the buffering of milk between pH 6.6 and 8.3. Titrable acidity is reported as percent lactic acid and is dependent on the volume of sample. The acceptable limits of acidity is less than 0.18%. Next is added water. Added water can be measured by changes in the freezing point of milk from its normal values. The current official freezing point limit is negative 0.505 degrees Celsius. The freezing point of milk is the constant physical chemical property of milk, which is determined only by its water-soluble components such as lactose and salts. The freezing point can be lowered by acidification of milk, which leads to protein denaturation, while adulteration of milk with water will increase the freezing point. The method can be used to measure the freezing point is cryoscopy. It is the determination of the lowered freezing points produced in liquid by dissolved substances to determine molecular weights of solute. Next is fat. The average fat content of raw milk is approximately 4.4 gram of milk fat per 100 gram. And the Gerber method is a historic method used to find the fat content of milk in particular milk powders. As shown on the slide is the butyrometer. Actually, the Gerber method is when the fat is separated from the milk by the addition of sulfuric acid, and then the fat content will be read directly on the butyrometer. For proteins, there are three different analytical approaches for the determination of protein quality. The first one is determination of total nitrogen, direct protein determination, and indirect protein determination. Dumas method is the current international standard and it uses chemical digestion. The advantage of the method is that it has high reliability and accuracy. As shown on the slide is the Dumas method. The milk is heated with copper oxide under carbon dioxide atmosphere, and then the milk is passed through the copper bed so that if any nitrogen oxide is formed, it can be reduced to nitrogen gas. And then the mixture is passed through potassium hydroxide to remove the carbon dioxide. After that, the nitrogen is collected.
Okay, lastly, we'll be moving on to the physical part of the process. So, I will divide it into two parts. First is for dairy processing stage, and the other one is for milk powder processing stage. First of all, I would like to start with the dairy processing stages. First of all, milking process. At farm level, the introduction of robotics such as automated milking parlours have been invented. The cows enter the parlour without prompting, and some cows are milked 3 times a day with increased milk product for the farmer. The tags on the cow allow for integration into the machine, which collect vast amount of data, including number of steps, chewing curd, and many more. Robotics milking machines have a lifespan approximately 13 years and then required further investment. Next, we will going to the separation and clarification process. Milk is then need to be clarified on the reception at the dairy to remove particles of dirt which will protect downstream processing equipment. Removal of bacteria, spores, and somatic cells from the milk can be achieved with centrifugation and also microfiltration techniques. As for centrifugation process, the aim is to separate the globular milk fat from the serum, the skim milk. This process is also known as skimming. Normally, it takes place at 122 to 140 degrees Fahrenheit or 50 to 60 degrees Celsius. Next, the milk will be going through a pasteurization process. Pasteurization was originally introduced to control Mycobacterium bovis, which causes tuberculosis or also known as TB. This process have been well presented in the slides before. Last but not least is verifying the pasteurization process. The test verifies the completeness of milk pasteurization by detecting alkaline phosphate, a natural enzyme in milk that is destroyed by the heat and whole time of pasteurization. As for milk powders, processing stages, there is only include 2 main stages, which is evaporation and also drying. As for evaporation, a falling film evaporator is commonly used in the dairy industry to concentrate the milk from 13% of total solids to a target of up to 52%. The evaporation is simply the removal of a solvent from a solution or slurry, and then it will be going to drying stages. When milk leaves the evaporator, it is passed through the spray dryer through small nozzles, which make small droplets or atomizing the liquid. The powder falls to the bottom where it is collected in a fluid bed under the cone of the drying chamber, where the fine powder behave in analogous manner to a liquid, and it can be conveyed without forming clusters.
That's all from us. Thank you for watching.
Bye.